go into the details of which type of sequence stabilize which type of fold. And we're going to build a quite simple model for this. So if you want to build a helix, what type of constraints do you have? You need to, right, form hydrogen bonds between adjacent amino acids. And you also need to have residues that like to be in an alpha helix. Yes? And so you need to have at least 4 to form one fold. And you need to have 8 to form 2 turns, and so on and so forth. For a beta strand, similar story, except the repeat of the pattern is not 3.6 to 4, it's 2, right? To come back to the same side of the helix, you need to have 2 amino acids. So the question is then, how long can these secondary structure elements typically be, given the constraints that we have in nature? So in other words, at which point does a helix or a beta strand break? What is the typical length of it? And for this, we're going to do a simple model, kind of typical of a physicist. So we're going to call P the property that leads to this secondary structure, to make the secondary structure. So let's say we call P the propensity of the amino acid or of the structure to form. And so here you would have 1, 2, 3, 4, 5, 6, 7, 8 elements with property P. And then you need to have 2 elements that are with property 1 minus P, right? Because if this was red, then you would have a longer helix. So the constraints that we have are a number of elements, R times P, and twice 1 minus P. And this will be then the probability of obtaining R groups in one stretch. You see that? Yeah? So this is our probability. And then we will calculate the average then length of this happening. And to do this, you build a weighted average. So you take your probability, the W of R up here, times R, and you normalize this by all the possible probabilities. You recognize the formula of weighted average? Yes? Probability times property, all summed, divided by all the probabilities. And we only take the terms that have R that are more than 2 or 0, that are more or equal than 2, because otherwise it's just randomness, right? So this is why you have your sums that start at 2. And if we take the formula we had for the probability from the slide before, it will simplify to this R times PR, and the sum in the bottom is the sum over the PRs. The 1 minus P's, you see, they dropped because they don't depend on R, and they're the same at the numerator and denominator. Okay? And then through applying the laws of series, which I won't go into details, like I won't do the derivation, and it's also not in the book. The guy tells you to just look up a math manual to the laws of series. You will find the following formula, ultimately. So the average length of such a stretch is then 2 plus P1 over 1 minus P. So if our probability is 0.5, then we will find that the average length of something like this is 3. Did you follow? Simple model. So this means that on average, then, what will happen is you will have blue, red, 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 blue, just because of the typical, the way the probabilities are set up. So does that sound familiar? Three what? Three residues are involved in a secondary structure? No? 3 is too short, right? It doesn't even form a alpha helix. So if you take actually 3 times the repeat unit of the alpha helix or of the beta sheet, which is 3.6 or 2, then you get values of 11 and 6. And this is typical of what you observe in structures. So it means that this probability of 0.5 that I was just talking about before, it's not the probability of one amino acid to be in a helix or not. It's the probability of 3 consecutive amino acids, 3.6 consecutive amino acids forming an alpha helix. And this you can check with bioinformatics, actually. So it is related to this hydrophobicity-hydrophilicity balance, but I'm just stressing that it's a different 0.5. Do you follow? Probability of, so this p equals 0.5 is the probability of 3.6 residues in a row to want to form an alpha helix, or in the same way, the probability of 2 consecutive amino acids to want to be in a beta sheet. Okay? Did you see what we did? We built this model, and then from this we inferred why, in terms of probabilities, it might be the case that we observe 11 residues in helices and 6 in sheets. You can actually do the same thing. It's not detailed in the book. I'm actually not sure how it's done, but apparently you can do the same thing for the average loop length, and then you'll find this formula instead. And this gives 5 if you take a probability of 0.5. And indeed, this is the typical average loop length. So this means that random sequences can easily form these layers that we talked about before, because they will have a typical property of folding into 11 residues helices and 6 residue sheets. So if you then put sheets in a nice way together or helices in a nice way together, you get a stable type of protein, globular protein domain. Yeah? I said this before, I'm going to repeat it still. Below about 150 amino acids, then random sequences will arrange in this kind of nice 2 or 3 layer globular domains. But if you have more than 150, then we get organization in separate domains. So now we have put quite a lot of constraints, actually, on how physics can guide the biological assembly of proteins. We know why they fold in small domains, and we know why the domains fold in the way they do, with pretty simple physics. Yeah? So now I'm coming back to this question of the Boltzmann distribution, and why does it look like we have something that obeys Boltzmann statistics, but instead we said that Boltzmann statistics can't govern this kind of assembly since we don't have exchange between different protein structures. So we'll talk about this for a minute. In fact, if you look at the statistics of small things, then they all follow kind of exponential type of behaviors. So this is an example of the occurrence of different amino acids that you can see here in the core of proteins with respect to the surface. This is on the log scale. And then here you have the transfer of free energy from alcohol to water. So let's say from something that mimics a protein to water. See how there's a kind of linear correlation. And because this is on a log scale, then there's an exponential dependency between the two, right? So this means that this kind of property, hydrophobicity and hydrophilicity of residue, their distribution in structures follows something that looks exponential. And a long time, we've actually noticed that this is true also for other properties, like rotamers in side chains of amino acids, occurrence of ion pairs, many other things. They all seem to follow exponential behaviors that look like Boltzmann. So bear with me, because I'm saying a lot of things, but I promise it will come into a coherent story in a minute. If we define native fold, we have to say that the native state is stable if the free energy is lower than all the other states. Yes, this will be the only fold because it's lower in free energy, and it has to be lower by at least kT because otherwise you have interconversion between states, right? Because of Boltzmann. And now we're going to consider a mutation from, let's say, serine to leucine. So from the plot that we had before, if we transfer a serine from oil to water, we will pay no penalty and we will gain nothing because serine likes to be equally, similarly in solution and in the inside of the protein. Leucine is a different story. Leucine, if you want to take it away from oil, put it in water, you pay a penalty, right? Because it's a hydrophobic amino acid, so it doesn't want to be in water. So when you take it from oil, put it in water, you pay 2 kcals per mole, roughly. Okay? So, so then the point is that inside of proteins, you will see serines and you will see leucines. But leucines will, when you bury it inside of the protein, you're gaining free energy, right? And when you're doing this, this means you're allowing other things to happen because you're giving energy to the system. So this means that maybe something else that is imperfect will be allowed to happen. So on average, you're going to see more leucines inside than serines, even though serines can be. Yes? You see that? So if we look at the total free energy that comes from folding, then the total free energy will be this delta F, and then we can sort of distribute this into small contributions. So the rest of chain plus the effect of the serine is delta F plus delta epsilon being 2 in the case of leucine and being 0 in the case of serine. And I'm building up to this notion. So this together needs to be less than 0 to fold. Otherwise, it prefers to be unfolded. Yeah? And now we have to look at the distribution of possible sequences. What are their energy of folding? And in fact, it turns out that their energy of folding, or free energy of folding, takes this kind of distribution. So it's Gaussian. Why is it Gaussian? It's always the same answer. Statistics? Central limit theorem? No? Okay. Yeah, so if you draw random sequences from everything that's possible, they're going to fall into a Gaussian type of a shape with average free energy delta F, which is the center of this distribution, and with spread standard deviation. So 70% of the sequences are going to fall into this interval, and it's going to be centered around delta F. The question is, where is delta F compared to 0? And in fact, it turns out that this average free energy of folding is way positive compared to 0. So even 1 standard deviation takes you still very far from 0. This means that a lot more than most sequences don't fold spontaneously. Okay? So the only sequences that will fold spontaneously are actually the ones that are below 0. And you can see from this distribution that this is a very small population compared to the ensemble of sequences that are possible. And in fact, the probability of folding is written up here, and it's the integral of this probability distribution taken between minus infinity and minus delta here, E here, which is written up there. Why is that? Remember, because delta F plus delta E has to be lower than 0. So I need delta F to be lower than minus delta E. Yes? You remember? This was the total energy. I need this to be lower than 0. So then I just write this as delta F is less than minus delta E, which is the same as writing delta F plus delta E equals is less than 0. So this means that this integral is only going to be this little black triangle down here, because minus delta E is then up there. So we can calculate this integral. And so this is basically kind of just integrating a Gaussian. So you can look that up. It will come down to this, because the Gaussian has an exponential form. Taking its integral will also have an exponential form. And what comes into the exponential factor then is delta E minus delta E over the variance divided by the average free energy. So this looks familiar. It looks like Boltzmann in the sense that it has E to the minus energy over something. Yeah? But you recognize that this is not KT. Instead, it's something else that we're going to talk about in a second. So now, increasing this delta E, delta epsilon, I should say, like in our leucine to serine, 2 kcal per mole, this is decreasing this probability really exponentially. Yes? So you just introduce a very small defect. 2 kcals per mole looks very small. But because the way the distribution is set up, you're really reducing exponentially the number of sequences that will then fit into this black triangle. Okay? So the key here is this exponential factor. Small defect, big effect because of the exponential. Small energetic defect, big effect. So we talk about this for a few minutes. First, what is this sigma squared over F? It replaces KT in the Boltzmann formula, right? So it has a meaning of energy. We can call it characteristic energy. And what's kind of interesting to look at is this sigma squared over F. So this is variance divided by mean. And you may recall from your statistics classes that the variance and the mean, they're proportional to the size of the sample, both of them. So the ratio of those is independent of the size of the sample. So this means that this quotient up there is size independent. So then the protein stabilization energy is not dependent on the size of the protein. It's only dependent on the magnitude of the energy that you lose from the defect. And this resolves our kind of question we were asking, why is it so bad to pay the price of one hydrogen bond? Because we were comparing this to the total energy of stabilization. But in fact, this derivation shows you that you shouldn't compare it to the total energy of the protein. You should compare it to the magnitude of the defect in an exponential format. Okay? So this means that a protein, whether it has 50 residues or 5,000, they have the same stabilization energy. Okay? But then it's much more likely to have a defect in 5,000 amino acids than in 50, right? And this also explains why proteins tend to arrange in small domains. Because in 5,000, you will have a lot of chances to make a mistake, to introduce a serine where you shouldn't. And so this nature never manages to make this happen. Okay? Right. It's a bit much to take in, but fingers crossed. The recording works and you can re-watch this. It's also kind of nicely explained in the book, so this should help you. So we can think about it a little bit also. Like we said, this looks like Boltzmann. So this characteristic energy, sigma square over delta F, we can think of it as the Boltzmann constant times a characteristic temperature, right? And this characteristic temperature will be the temperature of unfolding, right? And this is around usually 350 kelvins. It depends a little bit on the protein. So this means that these energy defects, they should be compared to kTc rather to the entire protein energy. And this is 0.7 kcals per mole. Kt with ambient temperature is 0.6 kcals per mole, right? So this is 0.7. So this means that a hydrogen bond that loses U5 kcal per mole, if you divide it by 0.7, it's a big number, and so it will not happen, basically. And that's why you can't accommodate defects very often. We good with this? Yeah, you don't feel like you have questions? Either you have a lot of questions or you don't have questions. It's a bit difficult to have, you know, a few questions, I think, in this thing. But pay attention to this derivation. I think it's quite cool. And so we're gonna finish this part of the lecture very soon with an example from biology. But I want to also ask you, do you know what's more common? Is it more common in the interior to have helices, or is it more common to have sheets? So you already saw it on the slides. I don't know if you were paying attention because it's kind of a lot, but do we typically have sheet with helices on both sides, or do we have sheets with helices in the middle? Remember the Rossmann fold? I'm going to take another vote. Who votes for helix in the middle? Who votes for sheet in the middle? Yes! You stopped voting.
Yeah.
Yeah, good. So why? Why sheets in the middle? So the reason is, or I don't know if it's the reason, but anyways, it can be explained by entropy. Sheets consume less residues per length, right? They take 2 amino acids to make one the same length than an alpha helix, roughly. So this means that you can arrange then the interior in more ways if you have smaller elements. And so this means that entropically it's more favored to have sheets in the middle than helices. There's just more ways to place 2 small blocks inside than one big block. So just entropic reasoning applying to sheet and helix occurrences in the middle of a fold. And as I just said, this is an example that shows that nature is complicated because it just does the opposite of what you think it should do sometimes. And this is GFP, green fluorescent protein. And here it's represented from the top. See how it's exactly the opposite of what we just described. It has a sheet that is arranged into a barrel, so better barrel, with a helix inside. So this means that that this can happen. And you know, it might just be like some kind of strange thing that you don't expect to happen from the principles we just derived, but it turns out this is a really, really important protein, in fact. It's really important because it's able to fluoresce. So this, in the middle of the in the middle of the barrel, up here you can tell, there's a small chemical moiety that's fluorescent. So it's called a fluorophore. It has this kind of shape. You know this, right? Conjugated rings, typically, they're able to fluoresce, so they can absorb and re-emit in the UV. And the original GFP, the one that you find in nature, it fluoresces as a green. It emits green light. It absorbs blue and emits green. But if you change the fluorophore, you can get different types of colors that are emitted. And this is really useful for biology because it's going to be really useful for biologists. But before that, it exists in nature. So this is a jellyfish that has a GFP that is makes its color. This is the guy that got the Nobel Prize for discovering GFP. Roger Tsien. And this is a bacteria culture that is made from bacteria that express GFP with different fluorophores, yellow, red, orange, and green. Unfortunately, that guy died really early. But before that, they showed how this could be really exploited. So I mentioned it's going to be useful for biologists because often we don't know what happens in cell, but with optical microscopy, fluorescence microscopy, we can see, where different types of proteins are located or something like that. But regular proteins, they don't fluoresce. But if you manage to attach a GFP to another protein, then you can reveal structures inside of cells. This has been really useful. And this is a more kind of application-based thing. You can't see anything. The point is actually that you can't see anything, but this is a little bit extreme. I think you really can't see anything. So this is ovarian cancer. And imagine you're a surgeon, and you have to cut tumors from this. So on my screen, it looks like what you expect, like a piece of meat. Like it's really difficult to see. But if you manage to tag the cells that have the cancer in green and you put them under fluorescence light, then you can tell the surgeon to just cut out the green from the black. This is much easier than cutting from meat. Imagine that. And not only that, but because we have all these variants of GFP, you can color in different fluorescent light different things. So you might not want to cut nerves, for example. That's what happens during surgery, right? You might cut nerves, and then you would cause damage to the patient. So you can color in different types of GFP, and then you can cut only the green, but not the blue and not the yellow. Okay? So this was just to relax you for a minute, talking about surgeons doing their work. Okay, so we have, according to my clock, about 15 minutes left. And we're going to spend those talking about how we get to the folded state. And I might not get exactly to the end, but then in that case, you won't have on the exam whatever I don't go through. So we discussed which folds happen. We discussed why they happened. And what we didn't discuss today is how they get to the place they are. So how do you go from unfolded to folded? And in Burke's lecture, you talked about kinetics of this, but I want to talk about thermodynamics of folding. Before I do this, I just want to show you a related example on a different kind of system. And yeah, bear with me. You're going to see how it's related. So for biology to happen, often we need things to bind. And there were two main models. So the kind of basic model that was around for a long time is this lock and key type of model where you have the protein and you have the ligand, and it builds a very nicely shaped pocket that is exactly the shape of the substrate. And the substrate binds, and then whatever else happens in biology. But who believes this anymore, right? It's never the case that you have this already perfect place for a key. Instead, then people came up with this notion of induced fit. And the idea was that you had a binding site that was vaguely in the shape of the substrate. And then upon binding, the protein rearranges around it. And then you get a tight binding in the same way as the first one. But so this is called induced fit because the substrate induces the protein conformation. And then it turns out that, of course, it's neither most of the time. And what really you have is a mix of both, and it's called selected fit. And so the idea is that you have a protein that can exchange between conformations according to Boltzmann statistics, right? And when a ligand comes, it will just shift the equilibrium towards the more fitting type of thing. This is called selected fit. So it's kind of an intermediate model where you don't go only that way, like induced fit would tell you, but also you get a population of proteins in the right conformation already in the absence of it. A real example of this is a glutamate binding protein. So in yellow is glutamate, and the unbound state is in red, and the bound state is in blue. You can see the blue will create a contact between the glutamate and the protein. And this actually happens through selected fit. And it's reversible, and the motion is pretty big. Was there a movie of this? No. Okay. Well, so this is an example of a real transition. A very interesting place where this happens is hemoglobin. We talked already about globin. Remember, it was this fold that was made only of alpha helices. You can see that hemoglobin is formed by 4 of these globins, and each of the subunit can bind oxygen. And they exist in 2 states, oxy and deoxy. Oxy is the oxygen-bound one, and deoxy is the oxygen not bound. Yeah. And why are we mentioning this? Is because the domains seem to influence one another. So look at this oxygen binding curve. In red is the hemoglobin one. And oh, by the way, the function of hemoglobin, like we said about the llama and stuff, is to bind oxygen and then to release it, remember, right? So this is the type of binding depending on the pressure that you get. You can see that hemoglobin is not a very good binder for a long time. So one lobe doesn't have a very high affinity for oxygen. But then when you bind one, it triggers a conformational change in the other that will already be in a state that is more favorable to bind oxygen. And then you get this kind of curve. See how it's sigmoid? A typical binding curve looks like the blue one. So you start with a really high affinity, and then as you're populating more and more the binding sites, then you get a lower affinity, right? So the red one is characteristic of cooperative transitions. Okay, now we have a movie. Good. So the reason I'm bringing this up is that do we or do we not have this type of cooperativity happening during folding, or the opposite of folding, which is denaturation? Just a reminder, for folding to happen, you need to have thermodynamic stability. We talked a lot about it today. We need to have a state that is very stable in the folded and that is more stable than the unfolded. And also, there's the kinetic problem, so you have to overcome this barrier. And I'm not talking about kinetics, because you already went and talked about kinetics, so I'm only talking about thermodynamics. And typically, when you look at gradually unfolding conditions, the type of signal you get is this type of sigmoid. So it's an abrupt change. It doesn't happen in one go, but it happens in a pretty steep way. So this looks like a cooperative transition. It looks like the protein goes from, in that case, folded to unfolded in a really sharp way. Okay? And just to give you ideas what causes unfolding, here is some chemical. I forgot this guy. But you get the same effect from pH, from temperature, from other type of unfolding agents. The first guy who discovered that folding was a reversible process is Anfinsen, got a Nobel Prize for this. And he showed basically that not only can you unfold, you can refold. So this is, I think, a CD spectrum, circular dichroism. I don't know if you know what that is. It doesn't matter. It's just indicative of secondary structure. And so you start at 20 degrees, the blue one. You go to 95 degrees, you unfold. And then when you go back to 20 degrees, you can refold. This tells you that it's a process that is reversible, right? And so now, as I was mentioning, we discuss this notion of whether it's a gradual or a non-gradual transition. So this is energy as a function of temperature, unfolded to folded, I guess. And you can see that you have something that looks like it's quite steeply dependent on temperature, which tends to be indicative of cooperativity. So all the packing hydrogen bonds coming together at the same time during the folding process, right? And in fact, this kind of curve is more characteristic of this kind of dependency. So this is probability as a function of energy at different temperatures. And you can see this type of process, you would have an intermediate population in energy, but this kind of curve is indicative of this kind of process. So you go from one state to the other directly without spending much time in between. Now, what you should see on these plots is that these energy distributions as a function of energy, they're not a peak. Or they are a peak, but they're not a sharp peak. So it's not like in quantum physics where you have one state that is allowed. Right? This is biology, so there's always a kind of broad-ish distribution of states. So the question is to quantify this type of broadness. Does this broadness mean that we have semi-folded states? Or does this broadness mean that there's fluctuations within the folded, within the unfolded? And to what extent do semi-folded states exist?
I have a question
Yes.
about that, actually. And this is related to the ending, and I'm sorry if I'm moving too much now, but what would ice and like to compare to something that we can understand better is like ice and water.
Yeah.
Because that is a gradual.
Yeah, exactly.
Process, isn't it? Yes.
Yeah, exactly. So typically folding occurs over 10 degrees, and ice to water occurs over basically 0 degrees. So in 1 degree, you're done. So this means that this is a steep process, but it's not as steep. So it looks like a primary first-order phase transition, but there is some spread in it, which we will try to talk about for the last 5 minutes. Okay? So calorimetry would be the technique of choice. I'm running out of time to really explain this, but basically this calculates the heat capacity when you're slowly increasing temperature. Heat capacity is energy over temperature, right? Dependency of change of energy or enthalpy with temperature. And so you can see that here you are spending some energy to heat the system, but then you get peaks which are indicative of a big change going on. And the big change is unfolding, basically. And you can measure this because if you integrate over temperature, then you get the total enthalpy that is associated with the change. Okay, so this will characterize quite well the temperature at which unfolding happens and the energy that you get from it. And so it does happen fast, but again over 10 degrees, not over 0 degrees, to be compared to what happens in water. And so the little exercise that we're going to do is we're going to try to compare whether this energy that we can measure from calorimetry is similar to the specific heat change that we would get from a melting unit. So we're going to do a similar kind of simple physical model that is similar to, well, in some way, simple physical model. And we're going to compare this to the specific heat that we measure from this exercise and from calorimetry. So if the melting unit is equal to the one of the full protein, then it means that the protein folds as one. And if it's lower, it unfolds a smaller part. And if it's higher, it means that it's many proteins that are folding at once. And to do this, we have to write the equation. So this is now, imagine we have just two states that exist, a molten state and a folded state. And where we can write the probability of being in the molten state using, guess what? The Boltzmann distribution again, right? So, so if we call G prime, the free energy of the molten, and G, the free energy of the native, then this is the molten divided by the sum of the Boltzmann weights of the native and the molten, yes? So we then just rearrange this to write it as the free energy difference, which is sum of energy plus or minus Ts, yes? This should be kind of simple at this stage. And then if we take our binding curves, which I said we can measure from experiments, then we can look at the variation of the probability of being in the molten state with temperature. And it actually, if you look at this, behaves as 1 over delta T. So this means I can compare this to the one that I calculate from this experiment. So here what I'm doing is taking, well, what I'm doing, or what the book is doing, is taking this expression for the probability of being in the molten state that comes from Boltzmann and deriving it with respect to temperature so that we can compare it to the experiment, yeah? And we'll come out with this formula, basically. So now, I think the slides are not written so well. So this is 1 over delta T, and it's also this. So then you can write 1 over delta T equals to this formula, yes? And now we will place ourselves where P molten is 0.5, so that's the transition state. Probability of being in the molten state is 1/2. And then in that case, by writing these two equations together, we find dP over dt from experiments will be equal to 0.5 squared, P squared, or P times 1 minus P times this. And then this means that we can extract the energy that it takes to melt 1 unit. Okay? From calorimetry, we get this delta H. We have the concentration of protein in our sample. So delta H divided by the number of proteins gives us the delta E for 1 protein. Yeah? Did you follow? So this is what comes from experiments, and this is what comes from our thought experiment of having a two-state model with a molten and a folded protein. And in fact, I don't have figures to show this, but I can tell you that if we compare these two quantities, then we find that small proteins that are below 150 amino acid, they obey this law. So they are a melting unit. So this means that a whole protein that is 150 amino acid, it folds as 1. And anything that is bigger than this folds as separate melting units. And this is related to what we were saying about the fact that these proteins are really organized in separate domains, yes? Yeah, good. Okay, according to my clock, I still have 2 minutes, but according to this clock, we're done. So I'm just going to tell you if you're curious, but I'll take a note that we didn't go through this. We said denaturation happens at high temperatures, and it turns out that denaturation happens also at low temperatures. Or could happen at low temperatures, but usually we don't see it because the water is already frozen. And this comes from the balance between between the energy and the entropy difference between the state, folded and unfolded, in the hydrophobic core of the protein. And so this is related to what I was telling you about the hand-in just before. And then because delta H changes with temperature, then you have a heat capacity that comes in. And so the free energy as a function of temperature, it's not linear. It becomes peaked. And so if you would be able to go below 0 degrees, then you would have these curves that go down. And this means that you can unfold the protein, not only with high temperature, but also with low temperature. And there's a reference there if you care to read about it. And this is why some proteins that live in organisms that live in very cold protein, they had to evolve the so-called cold shock proteins. So typical proteins unfold at low temperatures, but through selective selection, through natural selection, you can get proteins that are actually stable at low temperatures. And this is it for today, basically. You have your study questions.